Well, hello everyone, and a very warm welcome to this Cytel webinar on Estimands in Oncology. We have a great panel to talk with you today. My name is James Matcham. I'm a Senior Principal in Strategic Consulting here at Cytel, and I'm delighted to introduce the panel to you. But I think I'll ask the panel members to introduce themselves. So let's go in order on the slide, please, guys, and please introduce yourselves.
Hi, I'm Michelle Casey, and I'm an Executive Director of Biostatistics at Pfizer.
I'm Casper Ruvibach. I'm a biostatistician at Roche in Basel.
Hi, good day, everyone. It's Rob Hemmings, long-time regulator in the European system and now consultant.
Hi, it's Andy Stone. I work as a consultant statistician now, and for many years I led the oncology group at AstraZeneca.
And it's Paul Terrell. I work as a statistician within strategic consulting at Cytel.
Okay, thank you very much, everyone. So the agenda for today is that Casper's going to give a short presentation on the background to Estimands in oncology trials. And then we're going to have an open discussion. And the discussion is we've got a few questions that we have already. So I'll begin by asking one or two of those questions. But the point of doing the webinar is to address your questions. So if you've got a question that you'd really like to ask, this is how you do it. So type your question into the questions pane that you can see in the control panel in front of you. That question will come to me and I will feed that into the discussion as I can. You can send your questions in at any time during the presentation. And if we don't get to answer your question during the presentation, we will try and sweep them up in the few days after the presentation and get those to all the attendees. So if you do ask a question, it won't be lost. So we will try and address your questions as we go on through. Okay, so I think we'll begin with our agenda and we'll start off with a presentation from Casper. So over to you, Casper.
Thanks, James. So thanks again, everyone, for listening in. And I had the honor to open this panel with a short presentation. And the ask was pretty challenging to have 10 minutes to introduce the topic. And I tried to summarize a few key considerations and maybe key questions that can then also be addressed during the panel discussion. Next slide, please. So the first question I think that are on many people's minds when I talk to colleagues in the context of Estimands in oncology is what is specific to oncology when we talk Estimands. Next slide, please. One key aspect is that many oncology trials, the primary analysis relies on the time to event endpoint. And when we think how this whole Estimand debate, as I understand, came up is more from the angle of having endpoints that are longitudinally assessed over time. And there some challenges arose and then this discussion started. And the question is how specific is this to time to event endpoints as well? Next slide. In my experience in the field of oncology, there is also certain inertia towards implementing this whole Estimand guideline. And aspects that potentially contribute to that inertia is that in oncology, there is often a feeling that many of the endpoints we have are very standardized, that many of the methods that potentially come out of the discussions around this addendum have been used for a long time in oncology already. So there is a little bit the notion sometimes, what's in it for us in oncology? What's new with this whole thing? And if we go to the next slide, this is something that made me think a long time ago already. If you look into, and there are other examples that are very similar. When we talk about this notion of having a lot of endpoints very standardized in oncology, there was this work by Hoodis and colleagues where they, from published clinical trials, extracted the definition of DFS in adjuvant trials. And you see here, you have here a bunch of clinical events. And each cross means this is considered an event for DFS in that specific trial. And you see here, you have here 9 clinical events, and you can go through the table and you will find that none of these clinical trials actually have an identical consideration of these 9 clinical events. Although in every trial, this endpoint was called DFS. And I think the Estimands framework opens the door for a transparent discussion very early on in a trial. How should we actually count our endpoint, or how should we define our endpoint? Because some of them, or even most of these clinical events, potentially they're intercurrent events, and you need to think about how should we account for that. Okay, next slide. Another aspect is treatment switching, and I think in the field of oncology we are very used to treatment switching. I just give here one example, the RECORD-1 trial. You see here PFS, and then after the study was unblinded and filed for PFS, many of the patients switched from the control to the treatment arm, and later on, if you look at overall survival, you virtually don't see an effect anymore. This is crossover from the control to the experimental arm. In the field of immunotherapy, we often also see not just crossover to the other arm, to the experimental arm, but after progression, patients would actually take a similar medication that's already on the market, similar to the mode of action of the actual experimental arm. And that is yet again complicating then the interpretation of overall survival. Next slide. Another aspect that I think is something that hasn't been observed to this extent earlier on is, again, in the field of immunotherapy, sometimes blinding is not feasible. Now, if patients are randomized to the control arm, they sometimes have already approved options with same mode of action outside of the trial. So if blinding is not possible, they're randomized to the control arm, they will immediately leave the trial to get on another trial with a similar mode of action as the experimental arm in the first trial they were in. And then what you'll see is as a pattern like this, you have 20% of patients that were randomized to the control but have never been treated within the trial. And this proportion is just 1.5% in the experimental arm. And this is another trial, very similar pattern. Next slide. Rick Pechtour, the director of the FDA Oncology Center of Excellence, commented on this, and he found it quite bothersome. And he admitted that this hasn't been seen before, this extent of patients randomized but not treated, and all in the same arm. Next slide. So I think in these examples, the question is valid. We typically refer to the ITT effect. If we just take the patients as randomized, we look at PFS or overall survival, and then we evaluate that, and based on that, we make a decision about the trial. But is this really what we're interested in if we see something like this or the treatment switching example before? Next slide. Now I have presented 3 very, very short examples. And if you think about them, what do these examples have in common? Next slide. All these aspects of these trials that challenge our interpretation when we read them out, most likely you can anticipate them. If you are moving or if you are acting with your trial in a very competitive landscape, you know that there is a risk that if you cannot blind and patients are randomized to the control arm, that they might be tempted to leave the trial and try it once again in another trial where they have yet another chance to get on the experimental arm. Next slide. What I want to highlight with this is that a clear formulation of the objective of your trial is key, because then if you are aware of these features that can happen, just these 3 aspects I just described, and if you then sit down with your key stakeholders and start to discuss, okay, what do we actually want to show? What can happen if we start this trial now with the setup we are planning? And do we capture everything that can happen? Is the effect we are targeting with the trial now, is this really what we want? Will it help us in getting approval? And then also think beyond that. So a clear formulation of the clinical trial objective is the key. Start with the question you want to answer. Next slide. So if we think about the impact of the ICH-E9 addendum, we can summarize as follows. Next slide. If you think of this treatment switching example, pre-addendum, I think there would have been no question that an ITT analysis, basically just analyze everyone as randomized, would be primary. You file for PFS and then you don't see any effect on overall survival. And then I think very often this discussion comes up, oh, can we kind of rescue overall survival? Can we fix the thing? And there are methods that mainly have been developed in epidemiology. You might have heard of rank-preserving structural failure time models and similar models, where you can potentially try to find out what would have happened to OS if we hadn't unblinded and filed on PFS. This provides a few challenges. Most likely, or very likely, you would need data for these kind of ad hoc analyses that potentially you haven't collected, because you need data very often, depending on the method you're using, but you need data at the switch to model the switch if you want. And of course, all this is post hoc. Your first analysis of overall survival was negative, and then you potentially try to kind of rescue it with these kind of ad hoc approaches. Next slide. Now, post-addendum, if you anticipate that something like this can happen, you might be tempted to define a hypothetical estimand. So what would be my treatment effect in a world where no switching would happen because we filed a trial on PFS before? And I think before the addendum, it was very unlikely that this could have been proposed in a meaningful way to, for example, authorities. And I think it's not directly caused by the addendum, but I think the EMA has issued a Q&A document for this kind of questions. And I'm quite convinced that the Estimand language and the Estimand framework helped them to formulate that under certain circumstances, and these are still very, very difficult to meet, but now there is kind of a door open that such ad hoc analyses can actually be pre-planned. And we now have a language to talk about such kind of analyses. And if we do that up front when we plan the trial, I think the probability that we then also collect the data that is necessary to run these analyses is much, much higher. And also the quality of that data that we collect is much, much higher. In addition, the assumptions are made transparent up front. So we are proposing this hypothetical estimand here, and then we have kind of a starting point from which we can start a discussion. Okay, next slide. So this is just one very tiny example of the impact of the addendum, this treatment switching example. One very recent example where the addendum has proved its value is COVID-19. I think we are all now concerned with assessing the impact of COVID-19 on our trials. And the Estimand framework provided a very good language and framework, how we can start to talk about this in an efficient way. And if you see how quickly a few groups have provided guidance on how this can potentially be handled or which aspects should be discussed, I think that is directly related that we now have this addendum available. And if you're interested, here is one of the references that discusses the impact of COVID, or how you can assess the impact of COVID-19 on the objective and analysis of oncology clinical trials. Next slide. So in conclusion, potentially the ITT effect, as we traditionally call it, might not always be the effect of interest, but of course, you need to have very good reasons if you deviate from that, and the Estimand addendum provides you a framework and a language to transparently talk how you potentially would want to deviate. In general, I would say it potentially needs a little bit more time that you need to invest as a team, as a sponsor when you design a trial, but very often that time will pay off later, and it will open doors for you that were traditionally just closed, like with the hypothetical estimand for overall survival. So the Estimand framework, I see this very much as an opportunity for better designed trials, to make assumptions very transparent and then have a discussion about them, and not pretend we are doing the thing we have always been doing for the last 2 or 3 decades and pretending they're okay, but very often they're just okay because many of the assumptions are implicit and not spelled out. And I think the addendum will bring these assumptions more to the light. And I also see it very much as an opportunity for leadership of statisticians to engage your clinical team or your stakeholders within your company and beyond very early on to have a clear formulation of the goal of your trial. Next slide. And I think this also applies in oncology. So when I talked about inertia, I think the opportunities are also there in oncology. The next slide. And this is a shameless plug. You might have heard about this industry working group on Estimands in oncology, which is a European special interest group sponsored by PSI and FSPI. That is the official name. And also an ASA scientific working group of the biopharmaceutical section in the US. And if you're interested to join or just to want to learn more, then I invite you to go to the webpage, and there you can find all the details. That's it. Many thanks. And then I look forward to an interesting panel discussion.
Okay, thank you very much, Casper. Very interesting discussion. I'd just like to ask everyone to put their webcams back on so that we can see everyone, and we'll get into the Q&A. So we do already have some questions. And the first question is around whether the Estimand framework has added anything to the design and analysis of oncology trials, but in particular, has it added anything in discussion with the regulators? So I'm guessing, Rob, this is one for you to kick off with.
Thanks, James. Happy to start, and thanks, Casper, for the intro. So yeah, I should have put a conflict of interest declaration in my introduction that I was instigator and rapporteur for the ICH E91 guideline. And if I think back to mid-2017 when the draft was released for public consultation, there were comments coming back saying, does this apply to time to event data? Does this apply to oncology trials? And that was a bit of a surprise. Whilst it was true that the language was written more for longitudinal data, the issues we were trying to address with the guideline are absolutely relevant. For example, whether the lumping of data that are deemed less relevant to the research question with missing data makes any sense. And in oncology, of course, we have censoring, which is a mixture of intercurrent events and administrative censoring at the end of a trial. And critically, as Casper referred to, whether the measurements of data that might be taken after an intercurrent event are relevant to the research question. Indeed, whether measurements after an intercurrent event exist. After all, what is PFS apart from time to progression with death handled through a composite strategy, if you want to think of it that way? Beyond that, we've lived in a world where FDA and EMA have had different rules around developing a PFS endpoint. We have the issues to do with later lines of treatment, including, as Casper said, treatments that might come from the same class as the experimental agent. We have the almost particular issue of crossover from reference to experimental after progression. And we have the huge topic of what we do with quality of life data, which should be much more prominent in our discussions and in our regulatory decisions than they are at the moment, in part because of how poorly the research questions have been articulated and how poorly the data handling and statistical analyses have been conducted in the past. And of course, that is longitudinal data in the oncology setting. In terms of uptake, well, it's true that the ICH group didn't have clinicians represented. It was a stats-only group. And it's true that we didn't have direct links into the onc and heme division in CEDA, or the oncology working party at the EMA, although I'm sure colleagues were in the loop. I am no longer, of course, in a privileged position to know what the regulators are doing, but like Casper, I expect this is very much still developing. But I sense this coming in advices or in Type C meetings, SPA procedures, PMDA consultations, and I expect to see this coming in regulatory guidance. So, James, perhaps still baby steps at the moment, but this is coming.
Okay. What was interesting there was that you mentioned that the addendum was developed by mostly statisticians and not other clinicians. Do you think that's created a bit of a problem for us in practice as teams work with clinicians that perhaps don't understand it that much?
I'm absolutely sure it has. I, as much as anyone, have tried to explain this concept to non-statisticians, and it takes time, and it takes energy, and it takes effort. I think it's a wholly worthwhile exercise, by the way, once you've been successful. And those topics I made reference to, whether it's the different rules around the PFS endpoint, or what do we do with later lines of treatment, treatment switching, the whole discussion around the quality of life data, to be able to talk about what the research question is rather than just have a debate amongst statisticians about the methods, I think is fantastic.
Okay.
Now, would we have ever produced the guideline if the group had been also full of clinicians from multiple different therapeutic areas? I think people were waiting long enough for the guideline as it was, and so I accept that there's limitations to the way it was done, but I think it was probably a good decision, and it explains why the guideline's written as a language and a framework to have a conversation, rather than trying to give a definitive regulatory position on what the most appropriate research question is.
I think one of the problems is the terminology. We as statisticians can read and understand the terminology and it goes straight into our heads. But when I put this in front of non-statisticians, it's a blank. You know, they don't understand the words we're using. And when they don't understand the words they're using, it makes it harder for them to be involved in the discussions when we're discussing the topics.
I think that's fair, Paul, and I think you can say this should have been ICH E19. You know, it should have been divorced from E9. You can say it should have been a mixed or a mixed discipline group who were authoring it, and you can quite rightly say it should use less technical language. Actually, I think all of those things are surmountable in the end. But even once you get past the language barrier, it's still not easy to think in abstract conceptual terms about your research question in the context of intercurrent events that will happen. Even once you get past the language and everyone understands the language, it's not an easy exercise. Again, it doesn't devalue the exercise in itself. Perhaps it even makes it more important.
And I think that
and I shouldn't sound defensive.
Sorry. I was going to say, I think that highlights the importance of the statistician's role in sort of getting through that language barrier, right? Because I think what we've raised is a lot of the Estimands framework is not doing something different necessarily than what we've been doing in terms of our analyses, but making sure that you're bringing the conversation up front during the study design. So understanding when you're censoring, why you're censoring, and that the clinicians have that understanding and how that changes if you change those aspects, how that changes the clinical question that you're asking. And so, you know, maybe taking a step back and not talking about treatment policy and hypothetical and, you know, those kinds of things, but actually distilling it down to, you know, if you do this in your analysis, this is the question that you're answering.
I completely agree to that. And I think an important aspect is, while it is potentially a painful exercise to discuss this with your stakeholders and the clinical team, it is not that if you would not discuss it, the problems would go away. The problem will surface at some point, and maybe it will surface at a point where it will be even more painful, namely after you unblinded the trial and you find out you haven't collected all the data that potentially stakeholders would be interested in. So I really encourage everyone to talk about all this very early on.
And it was interesting what you said, Casper, about the leadership role for statisticians, and Michelle's just echoed that, and I completely agree. But at the same time, this is a little bit anecdotal, so apologies for that, but in one discussion at the Scientific Advice Working Party, a medical assessor said something like, I don't care about Estimands, I don't understand the words, but I really don't think the data after use of a rescue medication are relevant. Yes, brilliant. You've got it. You've absolutely got it. And what we're doing, actually, although we're in the leadership role, is putting the conversation back where it can be multidisciplinary, and you can talk about this between statisticians and clinicians. And we're not just talking about, do we like rank preserving structural failure time, IPC, or two-stage methods, and talking amongst a group of statisticians, which Paul, is a conversation really nobody understands.
Okay, I'm going to just turn us on to another question that's come up on the board for us. This is a question about treatment switching, and just to frame it a little, treatment switching from standard of care to active, and I guess the other way around, after progression is something that Casper talked about and is not too common, but common enough. How does the Estimand framework help us think about estimating treatment benefit in this space? And in particular, the question I have is it ever possible for a hypothetical strategy to be the primary Estimand in this space?
I can start. So whether that is, I haven't seen it done, but now I think with this, for example, with this Q&A document of the EMA, at least there is a guidance that even the hurdle is still very high, I'm sure. But now we have a framework to talk about this, at least. And in my opinion, or in my experience, before the addendum, this is not something you would have approached a regulator with. So I haven't seen it done, and I'd be very curious to learn about examples. But even if the door is maybe not wide open, it is now open, and in my opinion that is a step forward, but I'm curious to hear others on this point.
Okay, Andy.
Yeah, I mean, this is a big topic, this, but as a general statement, and I'm sure others have experienced this, there's a big disconnect in oncology for survival in particular between the questions folks want to answer from a trial and what the trial is able to answer, even in the situation where there's no switching. And what people tend to forget, I find, is that you don't randomize patients to a single treatment. It's not like other therapeutic areas where you follow for 9 months, say, or a month, and you compare the treatments. Here you randomize to 2 treatment strategies, which start with 1 treatment or another, or sometimes a choice of the control group. And the question, often I find myself, even in this situation, clinicians having to remind clinicians that the question we're answering is, does introducing your drug into the current clinical practice result in that population living longer? So even in the absence of switching, I sometimes find, I think people are trying to answer a question of what would have happened if, hypothetically, they'd have never received anything else, which of course isn't answerable, and I don't think is relevant either. But the situation gets worse, I think, when the switch into the experimental arm, either because you've got some prior data in which makes trial participants uncomfortable to not allow switching from the control to experimental, or in fact, as often happens, a drug's approved on a single arm trial or a late line, and we do a randomized trial to confirm that benefit and an earlier line, but of course then the control group can get prescribed the drug, and you can't avoid that switching. So in that situation, the only question I think you can reliably answer is one of sequencing. Is starting with your drug better than having the option to receive it later? Because that isn't the question we want to answer. The question we want to answer is, well, what would have happened had patients not been allowed to switch? And as we know, there's all sorts of different methods to do that. Some are better than others, in my opinion, but what they all have in common is some very strong and importantly, untestable assumptions. And it can backfire. I mean, I've been involved with the discussions when I was worked within a company, within a large company, which folks who worked in the HTA area, senior management committees, saying, well, you can do these analyses in the HTA. Why don't we just allow switching, and then that analysis can tell us what would have happened? So then I get really concerned, and this is a potential downside actually of the Estimands framework. Is it legitimizing, overly legitimizing analyses that in my opinion, can never be totally reliable, and in fact can backfire if people don't behave in the way you expect. You may end up getting a worse result, or certainly you may not get the result that you get is true. So I for sure, I think this whole Estimands, I've seen it in other TAs, incredibly helpful. This whole Estimands framework is helpful, and it enables a more informed discussion about exactly what question you're trying to answer. But with survival and oncology, I guess it often uncovers that makes easier to have the conversation that the question you want to answer is not the one that the trial is set up to answer. And as I say, just because a question is possible to try and answer doesn't mean it ever be reliable. And I would be really interested to hear from others whether you ever see, as the question says, a situation where, let's say an IPCW could be the predefined analysis that appears in the label, or whether it's only ever going to be reserved as supplementary. I mean, I'll nail my colours to the mast. I hope we never are in a situation where it becomes the primary, because I just don't see how that could ever be a question we could reliably answer. However, and I don't know to what extent other people have used it or it's been used, I could see some of these analyses being helpful in terms of trying to interpret the data at the end of the trial. But certainly I would feel uncomfortable if the results of some of these analyses ended up in the prescribing information.
So I think Andy's nicely rehearsed the issues around this, and I just wanted to emphasise the two levels at which the question needs to be considered. Firstly, whether it's the relevant research question or not. Very important. And actually, I would not say never to that, but I think we're in niches. But secondly, whether you can get a reliable estimate for that research question. And again, just to shine a light or explain the guideline, that concept is in there. If before an estimand is set for a trial, it's not just a matter of agreeing the research question that should be addressed, but having some confidence that you'll be able to make reliable estimation. And if you don't have both pieces, you should loop back and start again.
But you're also addressing, well, who are you trying to ask the question for? Because as we know, the regulatory question may be different from the HTA question, may be different from the patient question, what the patient thinks they want to know, not that we may ever be able to answer it. But that's the other beauty, is we can, who are we asking? On whose behalf are we asking the question?
Yeah, so I guess the thing I often say, although it's not universally true, it's difficult to get a drug reimbursed if it's not approved. So we have to be careful that, I mean, I agree with you, but we have to be, don't have to put the cart before the horse. You don't want to put things into your pivotal trials that are going to damage your approval. Now, of course, if you're going to get approval on PFS, maybe it's less of an issue, but within companies, there's the commercial decisions that go with that, is the ability to promote on a survival advantage, which of course is going to be a lot more difficult if you've allowed switching in your trial. So it has purely commercial implications as well.
Okay, thank you very much for that. I'm going to move us on to the next question that we have coming at us. And of course, doing a webinar at this time is never complete unless you're talking about COVID-19 at some point. So the particular question here is, COVID-19 has meant that some patients are just absent from observation for periods of time for many different reasons. What advice from an Estimands point of view could you give on how to deal with this when it happens in our oncology trials? And I'm going to put this one to Michelle first. So over to you, Michelle.
Sure. So, I mean, the impact of COVID-19 is really going to depend on many factors, including, where the trial is in terms of enrollment and length of follow-up, the study design, and potentially even the regions where the study is being done. And so I think that, in general, we should think about, maintaining the same objectives and analysis methods in the original protocol, but we need to consider the impact of COVID-19 on missing data and potentially intercurrent events. And the Estimands framework gives us a structure to discuss these impacts. And so I think we need to think about the handling of the pandemic-related intercurrent events, as well as missing data, and whether or not there needs to be modifications to the analysis plan to account for those, and whether that goes into the primary estimand or whether we need to consider instead whether we should do supplemental estimands or sensitivity analyses, depending on whether we're talking about intercurrent events or assessing the impacts of missing data. But I think certainly the Estimands framework gives us, a method and a language to discuss these and to figure out how we should make changes or additions to our analyses.
Yeah, Casper.
So one potential way to think about this is to say there will be a direct impact of COVID-19 on your trial, saying people will die of COVID-19 in your trial, and people will maybe stop treatment because they get treatment for COVID-19 infection. And in these cases, I think it's difficult to exclude it is not related potentially to your disease, to your tumor, or to your treatment for the tumor. So here one could argue maybe treatment policy is a good way of handling these kind of more direct impacts on your trial. If a whole healthcare system or a hospital is shut down, you can argue this is a more indirect impact on the patient. And potentially that can offer or can open the door to using maybe a hypothetical estimand in that situation, because you can argue it is just because the hospital is shut down. It has not, most likely it has nothing to do with the patient's disease or with the treatment for the disease, that this patient has now this intercurrent event that he or she needs to stop the treatment. So that is potentially a way to start to think about these challenges that arise with this pandemic.
It's really interesting that the addendum was put together before COVID-19. But what I've found is that that's actually just as well, because it's actually given us a framework for all the discussions that we've been having on patients that have, not able to come into clinic, or ICUs that have closed down and not being able to see patients, or even putting off treatment for patients as well. So I think in the absence of the addendum, it would have been quite a hard discussion to have had. And so I feel quite good that we've had the benefit here. I've got a particular question that's come up on the chat here. How would you handle a COVID-19 related death event for PFS?
So I'll be brave, and partly because it links into what Casper was saying, and partly because we can be a bit provocative here, can't we? Casper, I took a look at the paper, and I like the paper, and I like some of the discussion in there, and the concept which I've advocated in other conversations of saying there will be a world before COVID, and actually the world after COVID, whenever that may be, might not look that dissimilar. And so the research question that we initially set out with in our trial that's been disrupted is still relevant to the world in the way it will be. Now, there's this period in the middle where we're actually conducting the trial, where I have additional intercurrent events and I have some missing data. And by the way, there's a 1 hour semantic discussion you can have about whether we do have intercurrent events or missing data. But okay, let's not do that one now. A part of the guideline that I think is really helpful is this concept of granularity in how you define your intercurrent events. So now we can have treatment discontinuation for COVID-related reasons, treatment discontinuation for non-COVID related reasons. And if we want to go further, those reasons, that granularity can relate to the health status of the patient, or simply whether the site was closed or study drug was disrupted and the supply was disrupted. So we can articulate all of these different issues in a nice way. For the sake of being provocative, I would go back to our previous conversation and perhaps only in this situation say, I think you can justify a hypothetical strategy for all intercurrent events here, providing your data set's good enough to actually do the estimation that would then be needed.
Yeah, I mean, I'm just thinking about how I'm intrigued from others exactly what you would do here. I mean, I guess it's going to involve imputing some data for these patients based on the outcome of other similar patients who didn't drop out due to COVID and et cetera, et cetera. And these situations, rather like the treatment switching. So in the treatment switching situation, I quite like very simple analyses because it, particularly where patients don't switch in parts of the world or certain centres, a very simple analysis to say, let's do analysis in those centres where nobody switched. Because, I mean, there's some nuances to that, but that, you preserve the randomisation. That's, it's a good estimate of what would have happened without switching. So I as well as doing the imputation approaches, I would be interested, because clearly it's affected different parts of the world in different, I'm seeing that, different countries. Rerunning as randomised those parts of the trial, maybe those centres or countries where there was much less prevalence of these dropouts, and does that agree with what you get when you impute, which of course is then, to me, is more heavily laden on assumptions than one where you preserve the randomisation.
So I think that's where it comes in, where you have to think about whether or not you're actually changing your primary estimand versus doing a supplemental estimand or a sensitivity analysis, right? And I think you have to kind of take a step back first and not think, did I have a death due to COVID-19 in my trial? But do I have a bigger problem because of COVID-19 in my trial, right? If you have, a single death, is it, do you have to do anything about it? Is it really going to change the outcome of your trial? Unlikely. If you're seeing it, more rampant, and you need to assess it in a blinded way without looking at treatment. But is there a possibility that it could influence and bias the effect that you're trying to estimate within your trial?
Yeah, I think this is. I would also call for being pragmatic here, and very much in line with what Michelle just said. If these are very rare events, and I think I would anticipate in most clinical trials these direct impact events of COVID-19, be it COVID-19 infected patients or COVID-19 related deaths, and then we haven't really talked about how the death certificate is actually written globally. I think there is a large heterogeneity as well. So it's not so easy to say this patient died of COVID-19 with COVID-19, all these challenges. So I also advocate the pragmatic approach here. And of course, if you then have 20% of patients dying due to COVID-19, then it is worthwhile to start thinking about, should we change our primary estimand? But if there is any chance that your initially planned analysis and estimand remain halfway valid, even in this COVID-19 situation, then I would start talking with a pragmatic approach before I would start talking about more complicated, more involved approaches.
So for pragmatic, you're thinking potentially still more treatment policy type approach, and it's just part of progression-free survival. or overall survival?
I mean, if we think about if in a trial a patient dies in a car accident, you could also argue for PFS that should be hypothetical, because it is extremely unlikely it has anything to do. And if the patient was not even driving, maybe, then you can argue it doesn't really have anything to do with the disease or the treatment. And there we would also, I mean, typically we would say the patient died, you counted an event for PFS, that's it. And if you have one or two or three patients.
Yeah, I had an interesting question the other day. A patient can die from a car accident pretty much any time through a clinical trial, potentially, but a patient can't die from COVID any time through your clinical trial. Does that matter?
It does to me. I don't like the car accident example, Casper, because in the world post-COVID and in the healthcare systems post-COVID, patients will still die from road traffic accidents. Yeah, so this is still real life. The sort of precept for at least being able to justify a research question based around the hypothetical strategy is that COVID will disappear, a vaccine will be found, treatment effects won't depend on use of that vaccine, and so we have a basis to do something different. But again, it depends on the quality of the database to be able to do that, and that's both in ascertaining reason for death. And if you look at the statistics of death rates around the world, we can see how difficult that's going to be, and the rest of the data set being sufficient to inform whichever modelling, prediction or imputation approach we want to use. And so I don't pretend that's trivial.
Mm-hmm.
Okay, I've got my eye on the clock here. I think we probably do have time for one more question before we conclude. And the question was sort of almost asked pretty much straight away at the top of the webinar. Most statisticians deal with non-statisticians to engage in the Estimands discussion. How do we get non-statisticians and other people on the team to engage in an Estimands discussion at the design stage? How do we do that? I'm going to pick on Paul here to be up first for this one.
Okay, so I've tried this several times now, or many times, as I suspect some other statisticians have. You do, of course, have the carrot or stick approach. You could just say, it's here, there's an ICH document, you need to follow it, it's in practice. That approach pretty much failed. You can wait till they get the regulatory questions which are now coming in. The FDA, for example, pretty much always now puts an Estimands question in when you submit anything. So you could wait for that, but that's a little bit late. You'd rather get it in earlier. So I've tried two ways. One is a training approach where I sat down with them and I've given them an hour or two of trying to I make use of some of the ICH training, but I try to get the ideas across in a training session, try and explain the terminology, try and explain where we're coming from, and then move to their particular situation, which sort of worked, but wasn't great. So my preferred approach at the moment, and I don't know whether you've seen this, I saw it at PSI conference, James Bell from BI gave a talk on what he called detailed clinical objectives, and he took the objective of a trial and he split it. He just made about 2 or 3 paragraphs, but within those 2 or 3 paragraphs, it covered all the different aspects, the attributes, and the intercurrent events in a very top level. And you color code each bit. So the treatment, the summary measure, the endpoint, the intercurrent events. And if I just put that in front of the client, the sponsor, the non-statistician, and we start talking about each of those areas without using the Estimands framework, they start to realize why we're homing in on all of these different points. And at the moment, approaching it from what the objectives are and using this quite nice framework has been the most successful for me.
Okay, Casper.
I can only support that. So I was taught, or I'm still tasked within Roche to roll this out to statisticians and non-statisticians. And this is the approach you just described, Paul, is very similar to what we do as well. So you do it through examples, and maybe try to even not explicitly avoid the language, but make it very, very concrete. And one additional aspect that I found useful is make it therapeutic area specific. So if you talk to oncologists, then bring the oncology examples. If you talk neuroscience, then use an example from that therapeutic area. In my experience, that also helped. They then get more engaged and they know what you're talking about, and they know what potential intercurrent events could be. And yeah, we also kind of follow a little bit this detailed clinical objectives approach, and then try to convince teams to write that objective in their protocol, section 3 or so, I think. And then from there, everything basically follows from that. And you have to discuss these questions at some point. But if you do it up front, you have a lot of benefit.
Mm-hmm. Okay.
So I agree with what's been said, and whether we put it in terms of a more detailed trial objective, or there was a reflection as we were coming to the end of the drafting process that we should have just talked about treatment effects. It's just a more precise articulation of the treatment effect that we actually want to estimate. We should have just talked, or we could have just talked about treatment effects rather than Estimands. I think the other part we touched on earlier, it's an opportunity to engage the clinical team. What is it that you want to know? What is it that you want to know? And that's worked well. You have to be prepared. You have to have a sort of structure to take someone through a conversation. But putting the ball in their court and empowering someone to articulate what it is they want to know about the treatment, I've found to be quite effective. And the discussion we had earlier around difficulties in estimating treatment effects on survival in certain situations, and the whole mess around quality of life, I would have thought that was incentive enough for companies to want to engage in doing a better job with the data we collect in oncology trials.
Yeah, I guess just I mean, it's slightly off piece this, but you mentioned quality of life in your intro, Rob, and that really struck a chord with me because we do a terrible job with that in oncology, and people sometimes use MMRM and all kinds of stuff that's not really suited to it. And I'm a big advocate and had some success that continue to collect quality of life after progression. The interest in other folks' experience to provide some data. Do patients actually feel worse when they progress? Because if they do, you'll be able to demonstrate, you know, within the first 6 to 9 months therapy, your drug does better. Patients feel better in that period, by the end of that period, because you've delayed the progression, or you've delayed them going on to chemotherapy, and that'll get reflected in the poor quality of life. So that is definitely, I mean, it's not at the top of the list of questions for us in oncology, but I think that there's because the ITT framework is so embedded in oncology, I'm a little bit like the inertia you mentioned. Is can't we get, so what? We do this already. And working other TAs, I see the massive advantage it's got. You know, where the thought of collecting data after somebody discontinues, it's like, what? You know, why would you ever do that? But I do think the situation is very similar to quality of life, and this whole area gives us a massive opportunity to improve what we do with that data in particular.
But as you say, Andy, we don't do ITT or treatment policy at the moment. We often stop collecting data at the point of progression or when treatment's discontinued, and then we might analyze observed data, or we might use a mixed model for longitudinal data, and wow, what do either of those tell us, especially when we have different times on treatment, because we have a drug effect on progression of disease. And then it gets even more complicated if we want to try and address this wonderful concept of manageable toxicity. So I have 20% grade 3 plus diarrheas or GI toxicity or whatsoever. Actually, it might be interesting to use quality of life type data to try and assess the impact of that. And then, you know, treatment policy and collecting data after discontinuation doesn't make any sense at all. So again, we have to articulate the research question that we're interested in. Or I shouldn't say it doesn't make any sense. If you come from the comparison of treatment strategies basis, then of course it does. But if we want to learn about the adverse toxicology of a particular intervention, then arguably it doesn't.
So we just had one point on the questions that came in. It's one thing talking to our clinical team and educating them about Estimands, and clearly this is coming from regulators as well as the industry. How about journal editors? Would it make sense to be educating them to be expecting to see those presented in papers?
Why not?
Yeah, I think extending this to also more academic trials, I clearly see the benefit of the concept, and then you could incentivize trials run outside of industry you incentivize them with publications, of course. So yeah, why not?
Well, and you also need a common language across publications and trials to, put the whole space into context, right? So you may have a drug with the same mechanism, or even an academic trial or other trial that uses the same drug, and to try to put all of the information together to synthesize it, you need to be understanding what was done across the trials. And that's sort of the problem that was highlighted by Casper with the disease-free survival endpoint, right? If you look in publications, they say disease-free survival, but nobody knows what that means because it means something different for every trial.
Okay, I'm very conscious of the time and we're coming up to the hour. But before we go, could I ask each of you to give one piece of advice to the audience on dealing with Estimands in oncology trials? So have a think about your one piece of advice you give, and we'll close out on that. And I'm going to go in the order, on my order on the webcam, and it starts off with Casper. So Casper, your one piece of advice, please.
So see it as an opportunity to design better trials, make assumptions transparent, and engage stakeholders early.
Great. The next one I can see is Michelle.
So I would say don't think about this as something that is drastically different from what we've been doing before, but a way to discuss it earlier in the process to make sure that there's a common understanding that there is a synthesis between the clinical trial objectives and the analyses that we actually plan on doing.
Okay, Andy, you're up next.
Yeah, this has been said already and echoes the 2 points. These can be difficult conversations, and they can be, oh, it's the statistician making things more difficult for me. But have those difficult conversations early. I think Casper said it. It's much better than when you get results out and say, well, nobody told me that's what it meant. Why didn't you tell me that? And you may have even said you need to make people listen, but don't avoid having those difficult conversations. And you've got, if you like, something you can cite. Look, the regulators know about this. They've written a whole appendix. I found that really helpful, going back to the previous question. It's not something I'm making up. It's not some arcane thing. It's important, but always have those conversations early.
Okay, next up is Paul.
I would say understand the journey that people on with regard to Estimands. When I first heard about it many years ago, I was a skeptic. I thought, why do we need this? It's nothing new, it's pointless, it's all worked fine in the past. Through quite a lot of time, I've gone from a skeptic to a complete fan, and now it's just embedded in my head. With the whole COVID thing, Estimands COVID, it's just how I'm assessing it. But other people we're working with could be at the skeptic end of the journey and have not yet got the hang of it and are at the happy other end of the journey.
Okay, and last up, Rob.
Yeah, I mean, the framework is an opportunity, not a threat. And it's very gratifying that people find the framework useful. And I guess then I'm just asking for anyone who gets this, understands it, buys into it, should be the champion for someone who doesn't.
Right. Super. Thank you so much, everyone. It's been a great webinar with some really good questions. We have had quite a lot of other questions as well. We'll work over the next few days just to provide some answers, if we can, to those questions that you've spent time typing in. So thank you very much for contributing. And last up, I'd just like to thank all of the webinar panel very much for their time and for the contributions to the panel. So thank you very much, everyone. and hope you all enjoy the rest of your day. Bye now.